Let's get the time going.
This is DNA. DNA is made up of a sugar phosphate backbone with hydrogen bonds and nitrogen bases. And so each part of the DNA is made up of nucleotides, which are made up of a phosphate group, a deoxyribose sugar, and different nitrogen bases. And there are 4 different nitrogen bases. There's thymine, adenine, cytosine, and guanine. And adenine only pairs with thymine, and cytosine only pairs with guanine.
Topoisomerase relieves overwinding strain ahead of replication force by breaking, swiveling, and rejoining DNA strands. Helicase unwinds parental double helix at the replication force. DNA polymerase 3, using parental DNA as a template, synthesizes new DNA strands by adding nucleotides to an RNA primer or a pre-existing DNA strand. Primase synthesizes an RNA primer at 5' end of leading strand and at the 5' end of each Okazaki fragment of the lagging strand. Oh, and ligase joins Okazaki fragments of lagging strand on leading strand, joins 3' end of DNA that replaces primer to rest of leading strand DNA.
The first step of DNA replication is when DNA is unwound by topoisomerase and helicase. The second step is when the leading strand is synthesized in the 5' to the 3' direction. Then, the lagging strand fragments are synthesized. These are the Okazaki fragments. The final step is when ligase joins all of the Okazaki and the other fragments together.
The different DNA mutations are substitution, insertion, deletion. In substitution, there's point mutation, which changes only one amino acid in the chain. And then nonsense mutation changes the amino acid will end up coding for a stop codon. And then missense mutation, the change in amino acid will end up in a regular codon. And then silent mutation, the change in amino acid will end up in the same codon. And then insertion is a frameshift mutation which adds an amino acid, which can cause nonsense. Deletion will remove an amino acid, which can cause missense. And then there is a 3 nucleotide deletion, which can remove an entire codon.